Abdullah University of Science and Technology in Saudi Arabia. So Dr. Zhong got his PhD in Sichuan Agricultural University, and now he is working with Professor Wuhan in rice genomics by the next generation sequencing and the third generation sequencing. So his talk today will be pan-genome study of Asian rice. Dr. Zhong, are you online?
Yeah. Hello. Good morning.
Good morning.
Can you hear me?
Yeah, we can hear you. Please share your slide. Thanks.
Okay. Thanks for introduction. Could you please let me know if the slide is
great.
Okay.
Please go ahead.
Okay, thanks for introduction again. So my name is Zhong, as introduced, and my talk is going to be the pan-genome study of Asian rice. And Rod want to say express his apologize because he had emergency medical issue, so he cannot be here today. And this is our lab in KAUST, and we have a big team now. We have 5 postdocs and a couple of students, and we have some science specialists, and I'm here. This was our lab picture was taken 2 weeks ago. Okay, I'm going to show you our recent work about Asian rice. I'm going to start from our pan-genome of rice population reference panel, and then I will introduce the structure variation, especially for emerging index. And then lastly, I will talk a little bit about our variants calling, and including the SMPs and indels. But let's look at first part. So based on the 3K dataset as published in 2018 literature, and we have been upgraded the structure analysis, and we picked up one of the genome accession from the 15 subpopulations, and the largest are the mixed subpopulation. And we have been collaborated with Huan Agricultural University and Erie and Cold Spring Harbor. And we have selected these samples and do the genome sequence annotations and as a baseline data. And as we can see, the Lipan pair was published back to 1997 and published 2005 and then upgrade 2013. And also Minhui and Zheng were published from Huan Agricultural University, and they have really high quality genomes, and recently has upgraded to version 3. And other genomes, our genomes were not assembled yet. So we did the genome sequence and genome assemblies for the remaining representative. And based on those work, we have built the high quality genomes at Contig and 50 could reach 23 megabase, and Basco could reach more than 98%. And also we have averages of 18 gaps. So based on those works, we coverage all the subpopulations of the 3K Asian rice structure. So we all have these resources available at GitHub, and Persephone, that was a commercial tool, genome browser, and also have Grammarly site. We have all the genome sequence assembly available, and annotations available. And recently we are uploading our variants for each accession. And the next, I will go a little bit detail about the invergence. And as we know, the invergence is one important type of the structural variation, and it has been widely studied in many cases of species, and recently it had been published as several papers. In humans, they have been studied with a diversity panel with almost 300 genomes, but in crops and in rice, it has been widely reported as well. But normally, based on the third generation sequence, we have some special limitations of short reads. And also, based on the genome-wide alignment, we didn't carry out that a huge number of the genome assembly comparison. And based on this, rice has not been widely systematically studied. So we collected, including our MAGIC16 rice population reference panel, we collected the recently published genomes, including 29 PacBio genomes from Chen's paper that was published 1 year ago, and also recently published Oxford Lalipore genomes. And then we also upgraded 2 white species that is referred to Rufepogon and Punctata as outgroups. And based on all these 112 genomes, we filtered the genome quality again, but we kept the genomes only with count and 50 greater than 3 megabase. And also we found there are some large translocations, but without further evidence. So we filtered all of this with translocations or large absent deletions segment with more than 55 megabase. Based on the filters, we keep the 75 genomes in the following analysis. But before we move to the inversions with a large scale number of genomes, as we know, the inversion always be complicated, especially they have been highly overlapped with repeats, and also at the complicated at the breakpoint. We tested with different workflows, and we started with 4 workflows by using different aligners and different corners. And we found by using Nacmer as aligner and use Siri as a corner, we could identify as most of the inversions. And then based on this, we also did a couple of rounds of filters, and we first filtered based on the dot plot, filtered the negative, positive, negative, false, negative, positive. Okay, false positive results that was aroused by the tandem repeats. Then we further did the validation based on using the raw rates to support at the breakpoint. And then we also, by using the BioLand optical maps to validate the large inversions that greater than 1 megabase. And based on this, we can build a solid, powerful inversion index. And as mentioned, we have 75 genomes. Then we in total find and filtered get the high quality inversions 1700. And we also did the overlaps with in different accessions. We finally got 1400 clustered inversions. And we first studied the length distributions, and as we can see, the number of the inversions decreased with the length of the inversion increased, and the average length of this panel is around 50 kb. And here we only showed the 1 megabase inversions, and we have 11 inversions greater than 1 megabase. And we also studied the number of the inversions with the number of accessions we can use, and we found that by using 6 genomes are needed to identify the most common inversions. And then we studied those inversions, absence, and presence across these 75 genomes. And we could identify sativa-specific inversions and also outgroup-specific inversions. And we observed 323 punctata-specific inversions and 96 Hogan-specific inversions. And we also can observe 121 inversions that could be shared with Hogan and sativas that might due to the genetic inversions. And we also observed 873 inversions that's specific to the sativa. Then we estimated the inversion rates, and by using the outgroups and the inversions across the sativas, and we could estimate the inversion rate reach more than almost 64 inversions per million years if we compare to sativa to punctata. That is BB genome. And if we compare the replipogens, it could be reach 96 inversions per million year. But if we compare across only the sativa, it could be reach more than 700 inversions per million year, which is much higher than previously published. It's 2 to 50 times higher than previously published in 2020. But also in that papers, the authors also have mentioned that the inversion rate might be underestimated due to the quality of the genomes that were available. And then we further scan this in all the inversion index across the 3,000 samples of Asian rice. Here, I have to mention that the reason why we didn't directly use published 3,000 results of inversions, because that it was totally depends on the second generation sequence or larger platform. And we have discussed with the team a lot, and then we decided to scan the inversion index directly by using by looking at the alignment pattern at the breakpoint. As we can see there, we observed 4 main types of the alignment pattern. The first type is we can see the breakpoint was not obviously and is totally covered by the short reads mapping. And the type 2 is we can see the breakpoint, there's a sharp directly, very clearly. And this is the inversion breakpoint. And in the third type, we can see half of the alignment, and there's a breakpoint, but accompanied with deletion. And the last type is we cannot see any reads mapping to here due to probably a lot of reasons like deletions or sequence coverage in these regions. So we just include all of those results. And if we are thinking about study the 800 specific sativa specific inversion across 3,000 samples, we could got more than almost 2.6 million alignment patterns. How we can study this manually as well possible? So we used a machine learning approach with a detailed workflow like this, and we manually start the first part of the 10% of the alignment patterns, and then we use the machine learning to train the model by using these human studied alignment patterns and to predict the remaining of the alignment patterns. And as we can see, we can achieve the accuracy more than 95%, and the loss is less than 0.5%, less than 0.5. And based on these studies, we can learn the inversion index across the 3,000 samples. And then take advantage of that, then we started the detailed of frequency of all the inversions across different subpopulations. And then we further divided this in sativa specific inversions into genome specific inversions that could be observed in the limited number of genomes. And then we also observed 49 inversions that could be observed in one certain of the 16 subpopulations. And then we observed 37 inversions that specific to one of the major sativa groups. And then we also observed a large number, almost 50% of the sativa specific inversions could be shared across between different sativa groups. And even though with this group shared inversions, we still can see some phylogenetic evidence of the distribution of these specific inversions. And then we first studied the inversions, the effect to the genetic diversity and also effect to the phenotype. And we tested with 11 traits that was provided by from ERI. And we used the GWAS and also the linear regression models, but by using subpopulation adjust. And we found 32 haplotypes of the inversions that have effects associated with these 11 traits. And lastly, I will share some our recent result of the variants. And we mapped all the 3,000 samples to across our rice population reference panel, including 16 samples and also the reprogram. And as well, this is a large and computing resource required process. And we collaborate with our core lab here, and we designed an automatic GHK for workflow. Oh, by the way, the previous result based on 3K, that was GHK3. And then we also upgraded with GHK for workflow at 3 different platforms, that including the workstation and the cloud server and the high performance computing. And based on this automatic workflow, and we can do all the variance call for the 3,000 samples just in 5 days. And we also have applied our pipelines to the maize, soybean, and sorghum. And beyond that, we have developed our pipelines by using Docker and the Singularity, and this is barcode of wiki page, and they are all open resource, can be accessed, they are all available. And during early of this month, and we host a workshop of high performance computing workshop with a hands-on section, with a hands-on sessions in Thailand. That was a functional rice meeting, and I have led the project, and with our colleagues Naga and Manjuna, and we have more than 50 people attend the workshop, and this was a group workshop. This was a group of our workshop, and we are going to, because this is a resource required, and with the hands-on sessions, we found we got some surprise, and even this workflow could be run on the Windows platform. That was really surprise to us. And we have come back, we got the really good feedback from our audience, and we are going to host another online hands-on workshop next year, and we'll announce and send an emails out in the February next year. And by taking about the whole pipelines, we have mapped the 3K datasets to 16 reference plus reprogram. And we in total, oh, by the way, we can also extract each subpopulation specific database in that specific subpopulation. This idea was also recently published in the Human studies, studying a cancer disease. Title was multi-unstring GWAS exercise regarding that. So these ideas, I think we will have a very good, powerful data set based on those results. And in total, we identified 436 million sleeps with average 19 minutes per genome SMPs and average 2.8 minutes per genome in DELs. And most of our results, except the variance part, they were available in our 2020 paper and our recent submitted bioRxiv and paper, but that was got the major revisions, and we are almost fixed that. Hopefully, we'll go smoothly during the second round of the submission. And to brief conclusions, we have built this rice population reference panel, and all resources are available. and we detailed studied the pan-genome inversion index at the population level. We have explored a little bit about the well, for sure we estimated the inversion rate in Asian rice, and we are exploring the phenotypic consequence and the genetic diversity of Asian rice. Yeah, and lastly, I would like to thank our team again in KAUST and also our collaborators from Huan Agricultural University and the Professor Zhong and the PhD students Zhichao Yu have provided a lot of ideas, thoughts, and we do a lot of work together. And we also recently submit a lot of papers under review at in molecular plant. And also our collaborators from ERI, Dr. McLaney and Dima, and our collaborators from Cold Spring Harbor. Yeah, thanks for attention again. And I would like happy to take any questions and suggestions. Yes, please. It is quite nervous with present with a lot of professors.
Okay, please join me to thank Dr. Zhou for his nice talk. Is there any questions?
Hello, very good talk. Thank you very much. Actually, I'm very interested in the 4 different types of inversions. What's the biological consequence or biological significance for these 4 different inversions? Can you?
Yeah, thanks for your question. Yeah, this is a good point, and we are thinking, and we also need further thinking about that. So if they provide me the information of these different types, at least. For example, the first types, we can see that the resequence samples compared to the reference, they are matched very perfectly. They don't have the deletions. If we consider about the third one, they have a deletions at one side of the breakpoint. This provides us the information. Probably the inversions have their specific effects to the genetic diversity. And to be honest, we tested that. We combined the different types across the 3 samples, and we saw the evidence of the genetic diversities have been formatted or reformatted by these inversions. And we also see that from across 3,000 samples, the inverted samples and the standard samples, they have been classified from the phylogenetic tree to each group. So we didn't have that part to our manuscript yet. That's probably our next coming manuscript, but that provides us the information from the first 3 types. They are format or reformat the genetic diversities across whole genome wide. I don't know if I answered your questions, but yeah, that's a very good question, and we need to think and keep thinking about that.
Any questions?